I think it's now time to start. Good afternoon, everyone. My name is Carla Oliveira. I am the Secretary General of the European Society of Human Genetics, and it is my pleasure to be the moderator of the session today of this ESHG webinar series. Today we are on the episode 6, and we have as a guest and a speaker Serena Nik-Zainal. She's a clinical scientist from the University of Cambridge, UK. She's a professor of genomics, medicine, and bioinformatics, NIH research professor at Cambridge, and an honorary consultant in clinical genetics. So Serena is a person that has been working in the cancer field. She qualified in medicine from the University of Cambridge in the year 2000. She obtained a PhD at the Wellcome Sanger Institute in 2009, where she explored breast cancer using whole genome sequencing analysis. And her team and others actually pioneered the identification of mutation signatures that most of us use in our work today, either in research or diagnostics. And she has actually developed a lot of in silico and experimental tools to define these signatures and to allow us now to work with them as well. So she actually coined the term cataiges, and she was awarded a Wellcome Trust Intermediate Clinical Fellowship in 2013 and joined the Sanger Institute faculty team in 2014. So Serena ran a clinical project, Insignia, that was very important to gain biological insight into mutational phenomena occurring in people with DNA mismatch repair defects, aging syndromes, neurodegeneration, people exposed to environmental and occupational exposures. This is really something very important at the moment with a lot of attention. 2017, she moved to the Department of Medical Genetics at the Cambridge Center, and she wanted to accelerate the translation of her discoveries into the clinics. And that was, again, an important step for all of us working in this field. She was a member of the scientific program committee of the ESHG, and actually it was then that I met her and we had the opportunity to work together. It was beautiful times. And among the many awards that Serena received, I just wanted to highlight the 2024 ESMO Award for Translational Research. And it was given to Serena for the research in the field of mutational signatures and actually to the efforts of translating their use into the clinics. And this is also the topic of the talk today. So Serena will talk about recent advances in mutational signature and clinical applications. And we will have the opportunity to ask all the questions you want at the end using the Q&A box, not the chat box, the Q&A. Please list your questions, and I hope we can have a very nice discussion in the end. So we very welcome Serena, and thank you for joining us.
Thanks so much, Carla, for the very kind introduction and for the opportunity to present today. I'm going to share my screen, hopefully. Yes, I hope you can see my slides. Yeah,
yes.
Okay, great. So so thank you again for the invitation, and Carla, it absolutely was a pleasure to serve on the ESHG committee. Met a lot of people and made a lot of friends. So that was a really nice 4-year opportunity if anybody else on the call ever gets an invitation to join the panel. And today I'm going to talk about some of the more recent advances in the topic of mutational signatures, and then sort of our efforts to try to turn them into clinical applications. And up front, I'm going to acknowledge the young people in my team who've been doing a lot of the work. So in terms of scientific content, there's 2 parts. The first bit is on looking at mutational signatures of insertions, deletions, or indels. And that's been spearheaded by the 3 people you see there, Scott Nanda, Jean Ko, and Shujing Zhu. And this was a collaborative effort between the 3 of them. And in the second part, I'm going to talk about how we've taken some of those indel signatures and try to show you the clinical importance of them. And that is work that's been led by Daniela Black. And this is a manuscript that's in press, so please don't sort of extend this beyond the call. Okay, so just to remind everyone what mutational signatures are, these are imprints of mutational processes that have occurred through cancer evolution from the point of conception all the way to the point of cancer diagnosis and sampling you have all these mutational processes that have been happening, each one producing a characteristic imprint that we call a signature. When you sequence a cancer genome, what you get is a final portrait, and it's a composite of all those signatures added together. And it's a function of the size of these arrows as well. So if you've had a lot of exposure to tobacco smoke, for example, we can see quite a lot of tobacco signature. If you have some other repair defect that has occurred very early in tumor evolution, then it's going to be a dominant signature in the landscape. Okay, so mathematically, we are starting with whole genome data usually, and so that's going to be thousands and thousands of mutations. That's called our catalog. And from this catalog, we are going to try to identify the signatures, these pink boxes here, and then the amount of the signatures, which is the gray arrows. So those are called the exposures. So the catalog is the final whole genome product. The signatures are the patterns, and the exposures is the amounts of each pattern. And it's basically a linear approximation. So that image can be presented in this way. This catalog is just the counts of mutations. It can be any kind of mutation, whether it's subs or indels or rearrangements. These are the signature patterns. And as I said, here's the amount. And this should add up to 100% of the total numbers of mutations, right? So this is what, it's actually a pretty straightforward linear approximation when you're looking at one sample. When you're looking at many samples, it's still the same thing. It's just going to look a little bit more complicated in this next slide. But basically, this time we've got many catalogs. These are 20 different samples. They may still have the same 5 signatures, but the amount of each signature in each patient is a little bit different. So when you're starting with a catalog, when we are trying to identify the signatures, we call that an extraction. You're trying to extract the number of signatures and the nature of the signatures in your cohort. When we have the signatures and we're trying to decide how much there is in each patient, that's called an assignment. So that's estimating how much of each of these signatures is present in each sample. So you can see here in these breast cancers, these 20 breast cancers, these ones here are dominated by the green signature, signature A, and a bit of the pink stuff here is signature B. Whereas on the right-hand side is dominated by the yellow and the brown signatures. So that's just to give you a visual across the cohort. So, most of the work has been done on substitution patterns, single base changes. And this is just to show you some examples of substitution patterns. Signatures 1, 2, and 13 are associated with deamination either by methylcytosine or deamination that's been accelerated by apobex. And these are actually pretty normal processes. We have them happening in all our cells. By contrast, these are some examples of substitution signatures that are due to environmental exposures. So that's UV light, this is tobacco, and this is ursolithic acid. And these are very well known, very well described. Actually, well preceding whole genome sequencing and next generation sequencing, many others had been doing work in experimental models using specific gene reporter assays. So they knew what these patterns looked like. They didn't necessarily present it like this, but they knew what these patterns looked like. So when we came along and did the whole genomes and we started to see this, we could go back to their data to be able to say, they are exactly mirroring what other people had shown before. So all credit needs to be given to the people who started a lot of the work well before whole genome sequencing and next generation sequencing came along. Right. So that field has been going around now for 12 years. There's lots of people now in the field. There's so many different algorithms for identifying signatures. Every algorithm is as bad as the previous one. No best algorithm exists, I don't think, not even ours. But I would say this, it's not the mathematics that really matters, it's the biology that really matters. So it's how the catalog is constructed that matters. And to drive that message home, I'm showing you 2 examples here. These are 2 patients, and they both have tumors that are dominated by this red bar, C to T. And to all intents and purposes, when you look at this now, they don't look any different to each other. But if I presented it differently, I took these 6 mutation classes, and I now take into account the 5' and the 3' sequence context. So 5' ACGT, 3' ACGT, 4 by 4 is 16. 16 multiplied by 6 gives you 96. And I presented you exactly the same information, but in a 96 channel format, you're going to see that the signatures are different in these patients. So this patient up here has a largely signature 1. has C to Ts, but the C to Ts have tall peaks at CpGs because this is driven largely by deamination of methylcytosine, whereas the patient below has got largely C to Ts occurring at a 5' T. And this is the outcome caused by apobex. And so just looking at the 6 style context, we wouldn't have been able to see that biological difference. Whereas if you take the 96 channel approach, you can see it. And they're driven by these actually different biological processes. That is why it is so important to get the classification right. Okay, so more important than using the which algorithm is classifying the samples correctly. So that brings me to indels. So just wanted to demonstrate that with substitutions, but now I want to show it to you with indels. So for the longest time, for 8 years, because the signatures field started in 2012, for the first 8 years, we used this classification for indels, which is really crude, really simple. This is an image from my PhD thesis. And then from 2020 onwards, the Cosmic Sanger team came up with this classification, which is basically an expansion of the top thing, but including sort of the numbers of repeats for where some of these insertions and deletions occurred. So, for example, if you had a T occurring at a TT, then that's a repetitive sequence of 2. If it was TTT, then it's 3. If it's TTTT, 4, 5, etc. But what they did was they pooled everything after 5 or 6 polynucleotide repeat tracts. So when you do that, and we've got microhomology mediated deletions at the end here, but when you do that, what you get is these tall peaks because most deletions and insertions of a single base tended to happen at long polynucleotide repeat tracts. So although this was a sort of standard, people were finding it very difficult to do mutational signature analysis, including us. And actually, if you added all the tumors together that have ever been sequenced in the whole wide world, then you get this profile. This is 3,000 samples, and you'll get nearly all the signal, nearly all the mutations are in the same 2 channels. These are called channels. And if all your signal is focused just on 1 or 2 channels, it's very hard to do mutational signature analysis, or any kind of analysis for that matter. You just cannot see the biology and the granularity of the detail. Now, to prove this, we did a set of experiments. We used RPE1, H-start immortalized cells, and we CRISPR edited a variety of genes involved in the mismatch repair deficiency and polymerase pathway. So these are post-replicative repair pathway proteins. We either knocked out different mismatch repair genes, or we knocked in mutations in Pol E, polymerase epsilon, Pol D, polymerase delta. We also did some combined polymerase and mismatch repair mutants because we selected this because these are the things that we see in patients. So these were the ones we selected, and we simply asked the question, having grown them out in culture, are there genotype-specific differences? Are there gene-specific patterns in mismatch repair deficiency? And can we see them when we try to do mutational signature analysis? Okay, so we do the exercise, and just very crudely, we don't need to do anything clever here, we just count. So here we've got the wild type, that's our control. And then here are all our different genotypes, and this y-axis is on a log scale. So these mutations, mutants all have huge numbers of mutations compared to wild type. There's a couple that don't, but a vast majority of them have got a huge extra number of mutations. They most certainly have some kind of mutagenesis, which is what you'd expect for mismatch repair. Now, this is the image of the background mutagenesis for the model, right? So this is what wild type looks like. You got a big green bar and big orange bar. That's just our wild type. That's just what happens when you grow cells in culture. But when you look at the individual genotypes, what you get is this. So you see that all the signal is focused on just this 1 bar on the green and 1 bar on orange. There's a little bit of noise here and there, but otherwise, there are clearly differences between the different genotypes. There are some which are dominant orange bar, some that are dominant green, some that are only green or only orange. But it's quite hard to be able to separate these out well. And that is shown by asking the question, how similar are these signatures compared to the signatures that we pull out from cancers? So here we have a controlled experimental model. We know exactly what the genotypes are. We know that they're definitely mismatch repair deficient or polymerase dysregulated. When I look at cancers, how do they compare? And here are the signatures from Cosmic's the cosmic signatures for indels, because they have their indel classification. The mismatch repair signature is called ID7, it's in blue here. And a score means that there's a high similarity between our experimental signature and the cancer signature. And what you're seeing is actually all our mutants are not similar to the actual cancer-derived signature, allegedly, and they're all very similar to signatures 1 and signatures 2, which are these here, and you can see why, because signature 1 is made up of green bar and signature 2 is made up of orange bar. So all of these just end up being called as similar to these signatures. And these signatures are supposed to be signatures of normal. They're supposed to be just errors of replication that occur at a low level usually. And the actual mismatch repair signature that was allegedly identified in cancers is this one here, which I think we're a bit surprised by because this what was in here is missing peak at polynucleotide tracts of 6+ or more. This is where you expect to see microsatellite instability that's associated with mismatch repair deficiency. You expect to see a bar where there isn't a bar. And that's because in the cosmic classification, it's all being turned into ID2, which is why all of these are being called either ID1 or ID2 in this system. So we've proposed an alternative classification to try to enhance the ability to do indel signature analysis, because our sort of view is we want to try to get the best results for patients. This is all great for Nature, Cell and Science papers, but how do we make this workable for patients? We need to get it right for patients, and mismatch repair deficiency is one of those things that's targetable. So we've proposed a new classification. It's a little bit complex, but just to give you a high-level breakdown of it. So this is the old classification system, and this is the new classification system. There's a lot of similarities between them, but there are some key differences. And the first one is that just like substitutions, we have taken 5' and 3' sequence context into account. I don't know why this was never thought of before, but we've just done something that has been shown to be effective in substitutions, and we've put it into the indels as well. The second thing we did was remove the cap of the polynucleotide repeat tracks, where they had pulled everything that was 5, like TTTT and above, into one channel. We have spread it out to see whether we could see a bit more biology there. And the answer is you can see more biology as a result. And the third thing we did was remove any of the channels that didn't produce any signal. Because there are some indel channels that actually don't really produce any mutations at all. And so we've removed them because if they're not giving you any signal, they're just a way to reduce power in your analysis. So we've removed channels that don't have any signal. And this is what our classification looks like. Now this information is more spread out across all of the channels, and it's like 89 channels now. So although this is 89, that was 83, the numbers are very similar. Actually, they communicate quite different things. And as a result, you can see a lot more biology. So that's the MLH1 signal and MSH2, they look similar to each other. But they look very distinct to, say, these ones on the right-hand side or any of these POL-D and polymerase epsilon signatures. And now, because there's so much information coming from many more channels, when you do your analysis, they will come out as separate signatures instead of as just one signature or miscalled to another signature. Now, we did a lot of the mathematics as well to sort of show that the discriminatory capacity of the classification systems, how they compare to each other. So we performed a new signature extraction using our experimental models because it's a clean system. So we had all those gene knockouts and knock-ins. We know what the signatures are. And we asked the question, if we did it using the 83-channel system or the 89-channel system, how do they compare? And I know this is quite complex, and basically the solid and the dotted lines just represent different statistical pieces of information that we use to inform our decision on whether something should be called as having 2 signatures or 3 or 4 or 5 signatures. So the bar is the bit that tells you how many signatures we think is in that cohort. So using the pink cosmic 83 system on our experimental data, we think that there's 2 signatures, which is what you're seeing here, which is very, very difficult to separate. And we know we've got about 9, or we've got many different genotypes. We're expecting many more than 2. When we use the 89-channel system, we get 4 signatures in blue. which are these ones here. So the ones that are dominated largely by the yellow stuff or the brown stuff or the brown and blue stuff. Okay, so then we said, okay, now we're going to apply this to just a completely different cohort. Now we're going to apply it to cancers. And we applied it to a colorectal cancer data set. And this time we changed the algorithm. So we've kept the classification systems the same, but we've just used all sorts of different algorithms because there's 11 of them out there. We used ours, we used the one from the Sanger, we used the one from Harvard, we just use different ones to basically show systematically the pink bar is always to the left of the blue bar. So the blue bar is almost always finding more signatures, regardless of algorithm that is used compared to the pink bar. So that's really just trying to communicate that it doesn't actually matter what the algorithm is, it's how you classify the indels that matters. So we take this classification system and we applied it on several different tumor types. Here there's 7 different tumor types to show we identified basically 37 different insertion deletion signatures, and this is just how they're distributed across the cancers. I know that's too much information in that slide, but I just want to show you what these signatures look like. I certainly don't have the time to go through every single one of them. I just want to highlight some high points. So there's 9 of the signatures that we can map to the previous indel cosmic cohort previously, that we can map to their system previously. So they are basically the same ones, just presented in a different way. We found an Appleback indel signature. So, Appleback is one of the first things I described during my PhD. I'm slightly horrified that that's an indel signature, which I could not see. I've been doing this for 12 years. I'm amazed that it's taken us this long to see it. And we couldn't see it because we didn't classify the indels properly. Now that we classify them properly, we can see the Appleback signature. So I know it's humbling. I'm always learning. We've got these signatures that are due to environmental exposures and endogenous processes, and then these ones, which are due to post-replicative repair or PRR dysfunction. So that's mismatch repair and polymerase dysregulation. So it's not one, that was what Cosmic found. It's actually 8 that's actually present in the cancers. But that's handy because you saw the experimental data and now you're seeing the cancer data. And what we can do now is create algorithms to help us stratify patients more effectively. And we know that mismatch repair deficient and polymerase dysregulated cancers tend to be sensitive to immunotherapies. So using this new indel classification system, we trained a machine learning model. We used data where we knew 571 samples where we knew the ground truth. So we knew which ones were mismatch repair deficient, either by immunohistochemistry or by driver mutations or methylation, etc. We trained and tested the classifier on the 571 samples, and we created an algorithm called PRR detect. So some of you may know our old work for HR deficiency. That one's called HR detect, and this one's called PRR detect. And basically, it comprises combinations of substitution and indel signatures. So not one or the other, is both of those things. They'll have different weights. And we also take into account the ratios of indels to substitutions. And it's a multinomial classifier. So it doesn't just say whether something is mismatch repair deficient or not. It says whether you're mismatch repair deficient in purple, whether you're polymerase dysregulated in orange, or whether you're a combination of both of those things in blue. So that color scheme is going to be carried through. We now test this algorithm on a new cohort, and this new cohort comprised colorectal, stomach, and uterine cancers, because that's where we see a lot of mismatch repair deficiency and polymerase dysregulation. And the purple, orange, and blue is what you're seeing here on the screen. But what I wanted to also communicate was in the dotted line, that is what you would have found if you do that test called tumor mutational burden, TMB. TMB is just basically a count, and that is what is used clinically at the moment to try to identify patients with mismatch repair deficiency. And what I'm trying to communicate there is the lack of specificity if you use TMB. So here we've trained our algorithm. We know it's very, very precise. And I'm just contrasting it to things like TMB, which is going to give you a huge number of false positives. MSI-seq, which is another commonly used algorithm, or relatively well commonly used algorithm. It will find the mismatch repair ones, miss some of them. It won't see any of the polymerase dysregulated ones. MMR detects one of our first algorithms that will only see the mismatch repair deficient samples, but not the polymerases regulated. Now, if you use drivers, because everybody does targeted panels, if you just use drivers, you're going to see some of them, you're going to miss a whole bunch of them. And you might also have some false positives as well. So this is just to give you that sense of how PRR detect performs relative to some of these other algorithms. So just to summarize that section, that was a very fast gallop through a paper that came out earlier this year. But basically, I hope I've shown you that what's really important is actually how you classify the mutations, not what algorithm you use necessarily. And to show you that importance, we used experimental systems in human cells to show you that there's gene-specific differences. And then to show you how we have made the changes and then shown its value both computationally as well as applying it to cancers. We've also created this algorithm called PRR detect. And that's generally how we work. We do the experiments, we look at cancer data, and then we create the algo and sort of test it across different places. What I want to do next is show you how that value is being translated to other cancer types now. And so this is a piece that is in press right now. And this is being, again, I'm not going to be able to tell you about this whole paper. I just want to show you where we've applied the indel signatures. But this is breast cancers that have been recruited from the whole of England, recruited from 13 different sites. They've all been sequenced in the same place and processed in the same way. And then they've all had their drivers, their germline variants, their signatures, and applied all our usual algorithms to it. And on top of that, our colleagues have been very kindly collecting clinical information, histological information, and then we've been able to link every single patient because they are part of the National Health Service, which is a public sector healthcare system, to the Office of National Statistics to get cancer-specific mortality data. So we know the outcomes of these patients in terms of their cancer over 5 to 10 years. So this is unprecedented for us. Like I do a lot of genomics papers, but I nearly never have outcome data like this. So this was unusual, and we were able to validate some of our data, some of our findings on cohorts that other people have created and put in the public domain. So thank you to all of those people who do that. And in the interest of time, I'm just going to say to you that the data is representative, right? Because this is a question you must always ask when people present this kind of large scale data is it representative of the general population or not? Because if it's a skewed thing, then it's kind of, it's not always that useful. I'm not saying it's not useful, but it's a bit less useful. But here, yes, the whole genome sequencing data is representative of the types of breast cancers that occur in the UK in general. And yes, the cancer-specific mortality data was representative. So we did a whole bunch of QC to check that that was the case. And then, of course, we did all the genomics. So we've got all the drivers. I'm not showing all of it here. I'm just showing some important ones. We did all the signatures. We did all the compound algorithms for all the nearly 3,000 cases. But most importantly, when we looked to see whether there were any genomic features that could help us prognosticate about patients, given that we now have their mortality data, was there anything that acted in a prognostic way that's similar to the sorts of things we use in clinic? So in clinic, we use things like age of diagnosis, size of tumor, stage, grade, that sort of thing. And this is just to show you how it compares with some of those features. So this is our hazard ratio. You're going to see some of the most important factors on this slide. I can't show you everything because there's hundreds of them. And so, of course, all the statistical corrections have been put in, multiple hypothesis testing has been put in. I'm just going to show you some significant ones. So just to orientate you, anything to the right-hand side is a poor prognosticator. Anything to the left is a good prognosticator. So good outcomes, poor outcomes. And what you're seeing here is so grade 3 poor outcome is a high grade, grade 3 basically, so poor outcome, which is what we expect. Now what you're seeing here, P53 mutations in your tumors is a poor outcome, worse than grade 3 in fact. Structural variation burden, rearrangements, very strong poor prognosticator. And then SPS 213, that's APOBEC. And indel 9, that's APOBEC. Those two APOBEC features are associated with poor outcomes. In fact, and I'm not going to go into it, but when we try to find the most independent factors and we create a new risk framework, it ends up using these three things: p53 mutations, structural variation burden, and ApoBec mutagenesis. There are some other things here that are interesting, including some things that are actually good prognosticators. And so I'm going to go and explain some of these as we go through. So this one here is, I'm going to focus on this group because signatures 1 and 5 are associated with normal cells, just normal activity in your DNA. As you sit here listening to me now, your DNA is mutating. That signature 1 and 5. IND 1 and 2 were supposed to be reciprocal indel signatures of substitution signatures 1 and 5. So I'm going to show that to you. So this is signature 1 and this is signature 5, and we've reported this one of some of the first signatures ever reported. And generally, tumors with a high fraction of signatures 1 and 5 tend to have a better prognosis. And that's because it basically means your tumor doesn't have anything else in it. It's just got the stuff from normal, and there's not a lot else going on in your tumor, and your outcomes are probably going to be okay. And they tend to be enriched in ER-positive tumors and low-grade tumors. So that's all good. When you look at the indel signatures classified in the new way, you get these two IND1 and IND2, which Cosmic had previously described as being reciprocal of each other. One was slips on the nascent strand, and the other was slips on the template strand during replication. But we found that the behaviors were a little bit different between the signatures, because remember, we don't just find the signatures, we also quantify the amount of the signatures, right, in each sample. And what we found was, so here's ER-positive tumors, HER2-positive, and triple-negative breast cancers. Those are the main subtypes of breast cancers. And then you have grades 1, 2, 3, 1, 2, 3, 1, 2, 3. And what you're seeing is there's more of these benign signatures 1 and 5 and IND1. They track each other. They look very similar to each other, and they're mostly in the grade 1s sample. So they get less in the grade 2 and 3 because other signatures are starting to come on into grade 2 and 3. So as a proportion, you see them getting less. By contrast, for IND2, you see them getting more. Now, we don't have very many samples in this group in HER2, but in general, in all the other groups, they're not at the same levels as IND1, and they're getting more as you go through the grade. So they're behaving differently. So there were some indicators that this might be actually a different process. It's not just nascent and template strand slippage. And it had been postulated that it might be due to cell proliferation. And in fact, when you look at all the other tumor types, when you look at other cancer types, you do find that the cancers that are associated with a high proliferation rate tend to have much more of the dark purple IND2. And then if you look at the transcriptomes, because remember, many of these samples also have transcriptomes and methylomes, etc., which I don't go into much, but what we do have some of that data, we do find that the proliferation markers tend to be higher in the samples that have a lot more IND2. So we think that IND1 and IND2 are actually not reciprocal of each other, and they communicate very different things prognostically. So signatures 1 and 5, substitution signatures 1 and 5, and indel signature 1 are indeed very similar and probably entirely benign. And that's why they're the purple line that does well over time. So if you have a lot of that signature, you tend to be fine. By contrast, if you have IND2, you tend to do worse. Now, fair to say that the separation of the curves are not massive, and that's because in general in breast cancers, people tend to do well. But this is just to sort of communicate some of the biological insights that we're seeing from these signatures. Okay, the other one to point out is the ApoBec signatures. Remember I said the IND9, the indel ApoBec signature, we hadn't seen it in the past. We had only seen the substitution signatures, which I showed you earlier. And it's always been known that how you get this is cytosine to uracil deamination by ApoBecs, which when repaired incorrectly results in either C2T or C2G at this TPC sequence context. So we were a bit surprised to find this indel deletion phenotype that was occurring in our cancers. And indeed, when you look at the flanking sequence context, you find that this is the mutated C and is almost always downstream of a red T. And in fact, it tends to happen at a string, at a sort of a polynucleotide repeat tract of TTTT. And what we think happens is more or less the same thing as what happens in substitutions. You get cytosine to uracil deamination, but that occurs near a polynucleotide repeat tract. So when you're in the process of repairing it, you get something called an insertion deletion loop, and that sometimes gets excised and you end up with a deletion. So that's what we think, we postulate is the mechanism that underpins the mutational process that we're seeing. And again, also, as I said earlier, we're slightly horrified that it's taken us 12 years to even see these, but actually they add prognostic value to clinical stratification. And then last but not least, structural variation burden. In the interest of time, I won't go too much into that. I just really wanted to communicate this point about how we think when we treat our patients. So one of the classifications that we have always focused on is whether something is HR deficient or not, right? Because then the HR deficient tumors might get PARP inhibitors. So here's all the breast cancers, and 12% of them, that's the number, 12% of every breast cancer that walks in through the doors today will have a BRCA deficient tumor. It might not be from germline, it might be somatically acquired and then loss of the other allele, or promoter hypermethylation and loss of the other allele, or it might be hits in PALB2 or RAD51C and D. But 12%, that's more than 1 in every 10 breast cancers that walks in through the door is BRCA deficient. That is not the rate of treatment right now, and that is something that probably needs to change. So 12%. And by the way, this number has been corroborated by a separate cohort in Sweden. That's a paper that's still in review. My colleagues in Sweden have shown that 12% of breast cancers in Sweden have BRCA deficiency. So this has been now corroborated by another group. But of the 12% of the group that we call HR deficient, what I want to show you is that there's granularity there. So if I take this group and I look at this in detail, it's down here. So that's just a 12%. And what I want you to see is that there's a subset that's BRCA1 and a subset that's BRCA2. And actually, if you use the indel signatures, you can see differences between them. So this is your classic microhomology mediated deletions that underpins BRCA deficiency, and it's present throughout. Fair. That one we've always known. That one we've known for 10 years. But there is this signature in light blue, which is due to non-homologous end joining activity, we think. And it's actually got a different signature, which we could not see before. And it does differentiate BRCA1 from BRCA2. So it tends to be seen, it's enriched in the BRCA1 tumors, but not in the BRCA2. But look at this group right at the end here, which do not have the kind of microhomology-mediated deletions that the BRCA1s and 2s have. And in fact, they are none of these ones up here. They fall in this none group. And this is present, now we've seen this recurrently in many different cancer cohorts. So there's a subgroup in the HR deficient group, or HR deficient group. They're not quite HR deficient. In fact, now we've actually done experiments to show they're actually HR proficient. But they are, and they have a completely different indel feature. Actually, that's not true. Some of them are HR deficient, but that's not all there is to it. They have an additional problem, which gives them this additional signature. So the bottom line is, even when you think that something's HR deficient, there is granularity within that group that currently we're not utilizing. But I can tell you now that this group, because we're exploring this in great depth right now, we now know what the genetic causes are. There's multiple. And then we also know that what their synthetic lethality relationships are, and then what they're actually sensitive to in addition to PARP inhibitors. So that's just to sort of summarize our application of the indel signatures into the breast cancer cohort from Genomics England, and trying to communicate how we use some of those indel signatures as a positive or negative prognosticator, and how that might give us more granularity. So to finish off my talk, I just want to put everything in context, okay? Because and the way I'm going to do that is just to ask this question: what is in a breast cancer whole genome for a woman who walks in through the door today? And so of breast cancers that walk in through the door today, 27% will have some kind of genomic feature that is either immediately actionable or could go into a clinical trial very, very quickly. And that's mainly because some of these features are not things that you would usually associate with breast cancers. So you don't look for them. You tend to only look for the things associated with breast cancers, but actually they may well have potential treatments in other tumor types. And because we tend to regulate and we tend to say that you can treat this particular type of tumor with this drug given this mutation, what I'm trying to say is actually we should be treating the tumor based on its biology regardless of tumor type. So this is just to communicate that, it's a big proportion of breast cancers that when they walk in through the door have some kind of potentially actionable or immediately valuable information for clinical trial, which we're not fully using. Then there's another 15% in purple, which are signatures which we believe from experiments have translational research potential. So they could potentially, the base excision repair ones, these might be sensitive to PARP inhibitors. Perhaps some of these other ones have other sensitivities that we haven't explored. But if you did a whole genome, you'd know which ones they are. So you'd be able to find the right patients to your phase 1 and 2 trials. And then in this gray section, which is a bit more than 50% of cases, there will be the high-risk patients that we have identified through our prognosticator that I described in section 2 of this talk, that allows us to now identify patients that could go into trials for new treatments, CDK4/6 inhibitors, for example. So really what I'm trying to communicate is actually for every whole genome that you do from breast cancer today, nearly 1 in 2 cases, you could have some immediate clinical potential or clinical trial potential or translational research value. But this is where we fall flat, because at the moment we can do whole genomes in the UK, but we can't really act on it quite a lot of the times because we don't feel adequately informed. We don't have the evidence base to support its application. And so in the end, we end up doing targeted panels and then still doing nothing about it. And it's quite humbling to sort of realize what you can do versus what you do. And just to drive that message home, here are the kinds of breast cancers that we see such a lot of differences between them. These are the whole genomes of three patients. And the one on the left has got our BRCA mutagenesis. But these are 2 other women who are not related to her, and they have very similar whole genome profiles because their signatures are pretty much identical. Their signatures are the signatures of BRCA1 deficiency. How they have become BRCA1 mutant is different. One is inherited, one's acquired BRCA1, one is not genetic, it's epigenetic, but the signatures are identical. And what I think we're failing right now to do is to take all this information and to interpret and read whole genomes in a holistic way, rather than just say looking for one thing, one PIK3CA mutation or one BRCA1 mutation. We are failing our patients when we don't read the entire genome. So here are some examples of BRCA2 cases, which are, again, caused by different types of BRCA2 mutations, germline, somatic, and on the one on the right, it's not even a breast cancer, it's an endometrial cancer. And these are, again, very similarly looking tumors, but actually driven by other mechanisms, PALB2 and RAD51C, that all act together with BRCA2 in the HR pathway. Just trying to show you how different they look and how easily we can use mathematics to help us apply machine learning models and help to inform us as clinicians. So these are PMS2 breast, colon, ovarian cancers. Again, the signatures are very similar. This is called by PMS2 mutations, regardless of tumor type. This is mismatch repair deficiency, again, regardless of tumor type. I hope you can see they look so different to each other, to the previous genotype, but so similar to each other. And this is just really trying to drive home that value of tumor agnostic, holistic cancer genome profiling. And I'm going to end with plenty of time for discussion on this case, who is a 55-year-old patient. I have his permission to present his story. And he had an unusual undifferentiated pleomorphic sarcoma, which failed several lines of standard chemotherapy, and he had terrible disease progression. He was really quite sick. Well, we did a whole genome, and basically it looked like a mismatch repair deficient cancer. So this is a real story. And when you get a result like this and you feed it back to the clinicians, they go, okay, what do I do with this? I can't do anything with this. And that's because the guidelines are not there. And I think they feel like the legal frameworks are not there to support them. But actually, we all know that if you have a mismatch repair deficient tumor, this might be sensitive to immunotherapy. Anyway, it was hard, but he did get his immunotherapy. So these are his scans pre-treatment and then his scans post-treatment. And this man couldn't walk down the stairs, never mind up the stairs. He was so sick. But here he is performing. He's able to do a Bhangra dancing at his niece's wedding recently. So the improvement was really dramatic. And this is a single case, but there have been several cases like this now. But I think really the learning point was it's not even the cost of sequencing or even the analysis and interpretation that's an issue right now, but it is how we get the evidence base to support our clinicians so that when they get the report, they can do something about it. And so to do those clinical trials, what we need to do is change the mindset, Because right now, when people do a clinical trial, they look for one thing. Is it PIK3CA mutated or not? They ignore everything else. There is nothing else in medicine that we do that. We always take a lot of information into account when we try to make a diagnosis for our patients and to take them through care. So what we're trying to do in the UK right now is to change how we do clinical trial. And what we're suggesting, we have the infrastructure to do whole genomes, so we can do this. And I know not every country has it, but what we are suggesting is that patients are selected on a set of criteria. They get a baseline biopsy by whole genomes. So the whole genome is a triage step. And then patients are randomized either into standard of care, so there's one control arm, and then the rest go into genome-directed therapy, for which you can go into multiple different trials. Trial A, B, C, or D. These are different things. One could be HR, one could be pembrolizumab, one could be CDK4/6 inhibitors. They could all be different trials. But you're basically triaging upfront rather than having each trial do a separate and siloed investigation. So if you do it this way, we believe it to be more efficient. We'll get more patients into clinical trials faster. That is something we don't do well in the UK right now. And all for the purposes of trying to get the evidence base to support our clinicians so we can recruit and feed back that information so that clinicians can actually do something with those results. So I'm going to stop there. I hope that I've communicated the sort of the immediate clinical value that's in whole genome today, at least for breast cancer, and sort of highlighting that what we really need to do next is to get that evidence base to equip our oncologists to be able to act on genomics reports. I admit UK has got an unusual infrastructure, and it's a sort of place that could allow us to do some of these trials a little bit differently, but it will require a change of mindset to the one that we are currently used to in oncology today. So I'm going to stop there, and I'm going to thank the patients, the families, and all the clinicians that have contributed samples. This work required input from thousands and thousands and thousands of people, and my team and I have the privilege of being able to tell you about the data and how we use the data, but really we have leaned on an enormous number of people to help us get here, and of course our very generous funders. So thank you very much, and I'm happy to take any questions from the Q&A.
Thanks, Serena. That was a wonderful talk. And well, I learned a lot. I imagine that the over 100 people that attended the seminar will also have their questions ready. I think they are taking some time to write them down. So because I did write some questions, I will kick off with one. So you convinced us that having the whole genome sequencing analysis from tumors will help a lot, eventually addressing or guiding these patients to immunotherapy or olaparib or whatever other therapy that is adequate. Because I work with hereditary cancer, I have another question. Would you be able to screen tumors from patients that have missing heritability, so they behave and they look like Lynch syndrome patients or HBOC families, but they do not have a mismatch repair deficiency identified, that don't have MLH1 or MSH2 mutation or whatever, or BRCA or BR2. Could you actually, from the tumor, understand which germline variant is more likely to happen in those?
The great clinical question, actually, and something that I think has come up in our cancer strategy meetings in the clinic. So yeah, there seems to be families where it feels like there's some kind of genetic predisposition, but so far immunohistochemistry is negative, and can't seem to find anything on targeted panels. I think it is possible to look at the tumor to see whether there are any hints from the tumor, and then to also then look at the germline when you're doing that. I don't know whether anyone has done that systematically. We did have colleagues in Denmark. I won't say it was Denmark. It was Denmark. Mads Thomasen, I think, was the guy who led a study where he had families that looked like they were high-risk breast ovarian cancer, but no BRCA1 and 2 mutations. And he had a student who did the genome sequencing there, and actually, but it was a small study, and that's the only one that I've looked at that has been systematic. But they didn't find anything, as we said. We didn't find lots of new BRCA tumors that we missed or anything like that in that particular cohort. It wasn't a large cohort, but they hadn't found anything. But I think it's a good question, and I don't know. So I talk about signatures, but I don't think signatures are the panacea. I think we have to take all of that information together.
Mhm.
And have we always done a fantastic job of the germline? I'm not sure that we have, even though I'm a
Yes.
germline geneticist. I don't know that we have necessarily incorporated all the germline information to the best of our ability. So I think there's still more to learn.
Okay, great. There is a question from I don't know if this is Bill Newman, could be. He says, fabulous data and presentation. Thank you. For the 100K data, did you undertake interaction tests to show that the P53 SV burden and ApoBac profiles are independent predictors?
Yes, absolutely. Yeah, that was absolutely a critical thing to do. So they were independent predictors. And then we do all this complex mathematics, but as a clinician, we're all clinicians. You're there going, okay, I can't have a complicated thing. So we built a framework that was very simple to sort of help a clinician try to put patients into high risk, low risk, or intermediate risk. And basically, we were able to stratify the cohort really nicely just using these three. Some of the other things were not so independent, so we dropped them out. So we only kept those three things. It was exactly as you put it there. It's P53, SV burden, and ApoBex.
Mhm. Okay, I may have another one. So you know that there are patients, and sorry, I'm always jumping to the germline side.
That's okay.
But there are patients that carry homozygous variants in mismatch repair genes and
Yeah.
suffer from CMMRD.
Yeah.
So would you expect their germline cells, mainly the cells where tumors eventually will develop, to show some of the signatures you see in
Oh,
your
100%. So this exercise
the
has,
normal context?
yeah. Their normal cells already carry the signatures. So we've made iPS cells from blood samples from these patients. And if you look at their normal cells, they already carry mismatch repair deficiency signs. We did that in CMMRD caused by PMS2,
Mhm.
as well as MSH6. And yeah, you can already see the signatures from birth, basically. And what amazes me is that they don't develop cancers sooner. I mean, they do, right? They die of brain cancers a lot of the time from a young age. But it is quite interesting that every tissue has got mismatch repair deficiency from conception, and you can still form a human baby who will still grow up and only develop cancer at the age of 3, 4 years old. It is pretty astonishing how resilient human cells are.
Yeah. So I don't know if there are more questions, but I may have another one if in case somebody's thinking about. It's when your first data where you saw the profiles of the mutants with MSH2 alone or poly, I think it was poly, or the mix poly and MSH2. What it seemed to me, and this is a unknowledgeable view, it seemed that the poly signature overrode the MSH2 in
Mm-hmm.
the double knockout.
In the
You
doubles,
know,
the
yeah, it
combined.
seemed that the MSH was less relevant than the poly signature. Do you think that the poly has a stronger effect than the MSH2 in
So the
the?
poly mutants, just by sheer numbers, the poly mutants did have a very big effect,
Mm-hmm.
as big as a mismatch repair, but combined, when you combine them, it seems to be accentuated, like nearly an order of
Yes,
magnitude more
exactly.
mutations. But you're right, when you look at the shape, the poly features tend look like they were dominating.
Yes, dominating. Yes.
Yeah, it's true. I think that that's probably true. Now, we were expecting this question from reviewers, but we didn't get it.
Mm.
Um, and we were wondering whether it matters whether you get the polymerase mutation first before the mismatch repair or the other way around. Because of course that's what you're seeing in patients. You're seeing in patients having this combined thing. And I suspect that's what it is. I suspect, I don't have proof. We did try the experiment of
Mhm.
knocking one in first and then the other. But I suspect that if you have one of those first before the other, then the dominance of the signature is driven largely by the thing that comes first. That's my gut
Mhm.
feeling, but we didn't complete the exercise because we were not asked for it, and it was a lot of money to do the.
Yes, of course. Yeah. Okay, let's see. There's another question here. What about the germline variants that are in the high LD? Do you take this into account when structuring the specific signatures?
So your germline variation, whatever your germline variation is subtracted, right? Because we do sequencing of the tumor and sequencing of the normal. And any of your germline variation, whatever your haplotypes, is all subtracted away. So we don't even look at your germline variation. We're just looking at mutations that are arising somatically. So we don't really because it's not really relevant to what's happening somatically. We just kind of don't really take that into account.
There is another one. Do you have only had access to short-read whole genome sequencing data? Are you undertaking long-read to see if there are any hidden signatures?
So we haven't formally undertaken this exercise. It's an expensive exercise, right? To do all short read and all long read. Some people who have lots of money have done that. We're not one of those. So it was Isidro from EBI has done a huge exercise where he's looked at osteosarcomas or some kind of sarcoma, done short read and long read, and then said, what's the difference between them? And really, there isn't much difference between them, not at the scale of whole genomes where you're looking at patterns. When you look at individual complex structural variation, so let's say like a complex amplicon like HER2, right? You get copy number increased to 45, it's 45 copies of HER2 is being converted into an extra chromosomal DNA, blah. Then long read gives you information about the structure and precise breakpoints that have formed this amplicon. So that's the sort of information that you don't quite get with a short read, and you get higher resolution information from the long read. But in terms of general signatures, we don't seem to have lost information by using short read data. Yeah.
Mm-hmm. Yeah, unless this important information would be in the break in between the short reads that you would
So
be.
that's right. So we thought that that's
Yeah.
what we see. We thought that, or even in Isidro's paper, I think we all thought he's going to pick out all these things that we missed from the,
Yeah, exactly.
but
But it
actually
doesn't.
there wasn't anything.
Yeah. Okay, that's good news, right? We
That's
can
good
still
news.
work with
Yeah, I think so.
short
Yeah,
read sequencing.
yeah.
Okay, so I don't see any more questions. We are
Very good.
approaching
We're approaching time to
the
stop.
end. Yeah, so I would really like, on behalf of everybody that attended the seminar and also ESHG and Bill Newman, we actually launched the webinar series to thank you very much for being here with us today. It was wonderful, your talk and the explanations at the end. And I would also like to invite you and everybody that attended for the next webinar, which will take place on 29th of October. It will be Elfriede de Baar from Belgium, and we still do not have a title, but we are supposed to receive it later today, and so it will be available tomorrow. Your talk and all the other talks will be available in the YouTube channel. So if you want to look at it again or learn more or use it to your students, please do, because this was an amazing moment. So thank you. Thank you,
Thank you so
Serena.
much.
And
Thanks
thank
for
you
the
all.
invitation. It's been
You're
lovely.
very
Thanks
welcome.
so much.
Bye.
Bye bye.